So, freshwater watch. This is probably kind of teaching you to suck eggs, but it's kind of important context for why we felt we needed a citizen science water quality project. So obviously, water is very, very scarce, under huge amount of pressure, particularly fresh water, from kind of chemical pollution, but also abstraction, canalization, flooding, drought, you name it, it's all going on. And in the UK particularly, the last Water Framework Directive, 100% of our rivers failed chemical pollution tests. According to the 2016 State of Nature report, the UK is one of the most wildlife depleted countries in the world. And actually, globally, our ability to monitor and protect freshwater environments in a way that's consistent across the globe and can compare us in the UK to other people is quite limited. So enter Freshwater Watch. Freshwater Watch was created in 2012, and it was initially funded by HSBC. And the idea is that it's a global program to monitor the health of freshwater. And the idea is that if we can understand areas and causes of pollution, we can then help take action to improve them. So it's a global project. It uses a simple, really simple method that can be replicated by anyone, anywhere in the world, with some very basic training and internet access. So at a global level, every Freshwater Watch participant is collecting the same basic data. So we can compare data from Brazil with data from Zambia, with data from the UK. So we can start to analyse worldwide trends. At a local level, though, we also work through community groups. So we equip community group leaders with everything they need to apply Freshwater Watch to their own research questions and environmental challenges. And these can be university researchers, they can be catchment partnership hosts, they can be just volunteer groups, anyone who has a local freshwater challenge that they want to address. So, like I said, we started in 2012. We now have over 27,000 data points from nearly 30 countries. So I mentioned that we work through these community groups, and a really good way of thinking about Freshwater Watch is that, yes, it's a global citizen science project, but it's also a toolkit that any group can use. So group leaders have access to the Freshwater Watch method, which has been tested against lab samples, and the data has already been used to support over 20 peer-reviewed publications. So we're fairly confident in the data. They also get a platform and an app, and the ability to manage volunteers. There's customization options, so you can add custom variables to suit your project. So we have a lot of groups who, for example, want to measure invertebrates. You get a train the trainer, so somebody from Earthwatch will come and support you in how you do citizen science and how you train your volunteers. So that's a huge capacity building element. And then we also support our groups to make sure that the data they're collecting is put to best use, whether that be feeding data into policy, using it to plan restoration projects, whatever is relevant to that particular group. So the method itself is designed to be very, very simple, low barriers to entry, low cost, and to measure kind of a basic assessment of freshwater ecosystem health. So we've got some context, some very basic hydrological information, and some biological and ecological information. And all of that is just done using a simple set of visual questions. And then there are some basic tests for water chemistry, so nitrates, phosphates, and also turbidity. And what I'm going to do now is I'm going to play a video that will hopefully show the method in action. So if you haven't got your mics muted, please do that and cross everything that this works.
When you arrive at your site, open the Freshwater Watch app or get out your paper data sheet, record the visual observations it asks for, and take a photo. Using a well-rinsed container, collect your water. You can then use the measuring cup provided to measure the right amount of water. Then take one of the plastic test tubes, take out the pin, and squeeze out all the air. Put the tube upside down in the measuring cup and stop squeezing. This will suck up all the water from the sampling cup into the tube. The tubes contain chemicals that react to phosphates and nitrates to produce a colour. This reaction takes 3 minutes for nitrates and 5 minutes for phosphates, so you need to be ready with your timer. When the right length of time has passed, compare the colour in your tube with the colour charts provided. This tells you the concentrations of phosphates and nitrates to record. Turbidity is measured using a Secchi tube. Take your water and pour it slowly into the tube until you can't see the black and white circle in the bottom of the tube anymore. Then record the number on the side of the tube. And that's it. Upload the data using the app or the website, and you'll get an instant report explaining what you found.
So there we go. Now, hopefully I can, yeah, excellent. So hopefully you can see from that that it really is quite a simple and engaging method. Volunteers with very limited prior experience actually really enjoy being able to do that little chemistry test and see the colours changing. So it's really quite a great way of engaging wide audiences. But that's obviously not all we're doing. So what do we do with the data? How do we make sure that this data is being used? Firstly, at an individual level, we think the most important thing is to share the data back with the people who collect it. So we make the raw data available, but we also produce reports, videos, webinars, and other kind of different ways of engaging with people to explain the data. At the community group level, we encourage the community group leaders to use the data to solve whatever local issues they have. So as an example, we've got a group in Oxfordshire who've used their data to start a conversation with their local water company about combined sewer overflows and the impacts that those phosphates are having on the catchment. At high kind of greater spatial scales, so regional, national and international, we're feeding Freshwater Watch data into policies and management decisions. For example, working with the Environment Agency in the UK and also working with the UN to feed the data into monitoring progress towards the Sustainable Development Goals. And at Earthwatch, we quite frequently take this global data set and use it to publish in scientific papers and journals as well. So that's sort of what we do. That's our toolkit, and that's how we use the data. But what are the volunteers doing on the ground? So groups tend to use Freshwater Watch in two different ways. The first being a water blitz. So this is a kind of one-off event using a simplified version of the method. And the aim is to collect as much data as possible in a catchment over a weekend, and to engage as many different people as possible. And then some community groups also do more ongoing monitoring. So they have more committed volunteers who are investigating a specific issue. These volunteers sort of adopt a site and they go back month after month and almost act as stewards of the environment. So I'm going to talk you through kind of what we're doing with water blitzes and then what we're doing with community groups. So water blitzes, we're now getting quite adept at running these. They usually run on quite big spatial scales. So we've now done 10 events in the Thames River Basin district, which is what's shown on this map. And we've also run them in Paris, Luxembourg, Dublin, Sweden and Italy. And you can see from this map that a huge amount of data is produced. Each measurement is date and time stamped. And this is of much higher spatial resolution than can be achieved by professional monitoring. We also often measure sites that aren't routinely monitored, like garden ponds, for example. So the amount of data produced in these events is quite extraordinary. But also because it's quite a low barrier to entry, and we've got this trigger for people to take part, kind of go out on this weekend, we find that it's a really effective way of introducing people to some of the key concepts about water quality. So the pie chart is from our last Water Blitz report, and you can see that it summarises people's prior experience with environmental science before taking part in the Water Blitz. So we've got 21% who've never had anything to do with any kind of environmental work before, which is quite a high number for citizen science. So there's obviously huge value in the data. Recently, we've been working with the data in the Thames region to establish how these Water Blitz events can contribute to national statutory monitoring. The Water Blitz data give this sort of really rich snapshot in time of conditions across an entire region. And like I said, in a much greater spatial resolution. So this map on the left is one of the water bodies in the Thames. The black dot in the middle is where the Environment Agency monitor this water body. The orange dots are Water Blitz measurements. And then the coloured squares are the median phosphate concentration per kilometre grid square. So you can see that the Environment Agency are measuring in the location with the highest phosphate concentrations. And based on this, their Water Framework Directive assessment is that this is a poor quality catchment for phosphates. But actually the Water Blitz data shows that it's quite spatially varied within the catchment. So we're picking up information that's otherwise being missed. And then on the right, you can see what happens when we do regular Water Blitz events over time. We can start to take that kilometre grid square information, so very, very granular spatial scale, and look at how individual grid squares are changing over time. So what you have on the right, the black line with the grey error bar is sort of the 75% quartile of all phosphate measurements across the catchment. You can see that in these two particular grid squares, the green one and the purple one, phosphates in the green one occasionally are above what you might expect. And in the purple one, they are almost continually elevated. And neither of these streams are monitored by the Environment Agency at all. So alongside kind of doing that analysis, we've also worked with UK Centre for Ecology and Hydrology to create an online data explorer, which shows the Water Blitz data alongside Environment Agency data and also data from other citizen science projects. And I can put the link to this in the chat at the end, but if you explore this, you start to get a feel for how we've got two data sets that are both measuring the same thing, but both complementing each other. So Freshwater Watch gives this spatial picture, and the Environment Agency is much more dispersed spatially, but measures more regularly temporally. So the graph on the right is just two co-located Environment Agency and Freshwater Watch data points. The Freshwater Watch measurements have error bars on them, and the Environment Agency don't. So you can see that we've got, for those data points that exist, we've got quite good agreement, but Environment Agency are monitoring more regularly. So with our Water Blitz events, we've got this great complementarity. But then what about when we start monitoring regularly ourselves with these community groups? So this is then where we start to get groups who are actively exploring a specific issue. And we can work with them to tailor their monitoring to explore those specific issues, which is often not what the regulator or what the Environment Agency do. So we're looking at issues of local concern. We have hundreds of these groups across the world. So for this talk, I thought I would focus in on those that include farmers. So on our top, these are just 4 examples. On our top left, we have Qatar Flood. He is a farmer in Ireland who monitors impacts of nutrients on local rivers and lakes. And he's really interested in how cattle access to streams and timing of slurry application impacts water quality. So I'll talk more about him in a minute. On the bottom left, this is Matt Bruin in Oxfordshire. He's part of a group who are investigating sources of phosphate pollution in the river catchment. So this is that same group I spoke about earlier who were looking at combined sewer overflows. But Matt's particular site that he's monitoring is exploring the diffuse pollution element of phosphate pollution in that catchment. And he's now actively trying to reduce the diffuse pollution from soil erosion by using coppice bundles as a sediment trap. And Martha's been involved in measuring the nutrients and carbon that have been trapped in those coppice bundles, while Matt has also been monitoring upstream and downstream using Freshwater Watch. On the top right, this was a partnership between farmers and researchers at the University of Guangzhou in China, working to investigate the impacts of fertilizer application on farm water sources. And this was all tied into a sort of water stewardship scheme for farmers. So almost like a Chinese red tractor. And then in Madagascar, on the bottom right, we had farmers teaming up with local scouts to explore the impacts of small-scale subsistence farming on water quality. And the farmers actually ran a competition with the scouts to design the best solutions for reducing nutrient impacts. So that was quite a cool little project. So I'm not going to run through all of those projects, but I am going to show you a little bit of data from one of them, from Qatar. So as I said, Qatar is a cattle farmer in County Longford in Ireland. And he was taking part in a community group project that was originally investigating a lake in County Monaghan, but he couldn't get to his regular sites that he was monitoring as part of this community group because of coronavirus. So he decided to start a mini experiment on his own farmland. And he monitored small streams on the land regularly for about 12 months. And he did this in quite strategic locations to assess impacts of slurry application and cattle access. So this graph on the right is just one of his sample locations. And you can see he's measured really quite regularly over almost a year. On the first 2 spikes in phosphate concentrations coincide with him applying slurry to his farmland. The final spike is possibly the most interesting one. So he didn't apply any slurry to his land prior to this spike, but it coincided with the deadline for slurry application before the winter season. So across the wider catchment, you had lots and lots of farmers rushing to apply their slurry before this deadline. And so Qatar is now working with us on a paper about actually the policy implications of this, and is having these set deadlines negatively impacting these small streams. And as well as that paper, he's also adjusting his own practices. So he's fenced off some buffer strips to prevent cattle access to the streams. And he's also talking about neighbouring farmers about their practices as well. And he's now become a real advocate of citizen science. He really believes that citizen science can help farmers to better understand their local catchment issues and to become advocates for best practice. And that actually making this data and these methods available to farmers breaks down some of the trust issues that often exist between farmers and regulators, because farmers are able to see how the knowledge is being generated. And this is just kind of to give you an idea of why we work with farmers in particular, and why farmers are interested in citizen science. So Matt Izod on Bruin Farm, I mentioned earlier, with the coppice bundles. I wanted to take part in Freshwater Watch to see how our farming practices affect local water quality, as well as to understand the overall health of water around the farm. So echoing what Qatar said about actually having control of the data, being able to do this research for yourself rather than relying on external contractors to come in and measure your farming practices. And then Qatar, I never saw myself as a scientist. I don't feel like a scientist. I don't feel qualified enough. But when I put citizen in front of that, no problem. And I really like this quote because to me, it summarizes what citizen science is all about. It's about showing people that actually science and involvement in the environment isn't this big scary thing for people in white lab coats to do. It's actually this sort of collective responsibility that we all have for the environment. Citizen science offers people a way into that. So I think that's me done on fresh water. I'm now going to hand over to Martha to expand on the farmers piece, and talk a little bit about how we are now working increasingly with farmers on soil health as well as water. So Martha, do you want me to stop sharing my screen?
Is that... Yes, then I'll put slides up.
Okay, now I've got to hang on. I've got to figure out how to do that. There we go. Okie dokes.
Okay. Okay, so you should be able to see that now. Oops, hold on. Right. Okay, so I'm just trying to see my notes at the same time. Okay. So, can you guys all see my presentation now?
Yes, we can.
Yes, brilliant. Thanks, Izzy. Okay, well, thanks, Izzy. Do you know what? I know, obviously, I work at Earthwatch. I know about Freshwater Watch, but it's always really great hearing that kind of presentation and being reminded of what a great project it is. And as Izzy said, Freshwater Watch is very important for farmers. An area that we're working in increasingly as we've developed our sustainable agriculture programme over the last 18 months. Freshwater Watch could be described as a toolkit for citizen science assessment of water quality. And in recent months, we've been developing an equivalent tool for soil health designed primarily for farmers. We focused on soil health, and perhaps I'm teaching grandmother to suck eggs again, but we've focused on soil health because of the huge importance of healthy soil to agriculture, and also the vast potential for environmental benefits of management changes targeted at soil health. And yes, this soil health toolkit has been a key project that we've been developing in recent months. So I should say before I go into any more details of this, that when we say toolkit, there's actually very little, even less than in Freshwater Watch, that's physically provided. Everything that we'll provide for the toolkit is actually in that bottom right-hand picture, which is the only bit of kit that isn't that people might not have to hand. The toolkit is actually a set of instructions for assessing various aspects of soil's physical, chemical and biological health, using 5 very simple tests that, as I say, don't need specialist equipment. At the moment, we're very much in the development phase of this. We're focusing on developing the component tests, but we are aiming to eventually create an app that will have capabilities for data storage, data viewing and data sharing, as well as a function for discussion between users. So the aims of the project are to encourage farmers to increase their soil health knowledge through just hands-on soil health, we've been really surprised when testing the toolkit with farmers out in the field with how few of them do this on a regular basis and how powerful it's been for them just getting their hands dirty. The toolkit also provides objective data for farmers, and I should make clear that the aim of this is to complement, not replace, lab soil testing and working with agronomists, but it's giving farmers a chance to get to grips with their own soil health and understand what the data actually means. And that piece about perhaps not so much trusting regulators, it's really important for farmers to, I think collecting data yourself is very powerful. Ultimately, we want the toolkit to be within an app that allows data sharing, opt-in, I should say, data sharing, that could promote learning between farmers. So that could be local groups within a catchment, for example, or it could be communities of practice. So maybe grain farmers in a particular region could share data and show and discuss perhaps how different management practices are changing what they find. I've mentioned the positive environmental impacts of methods that are there to promote soil health, and we want to support farmers in minimising their environmental impacts. A great example of this is how, for example, if farmers are aiming to protect soil through increasing crop cover, through increasing vegetation cover over the soil. They're increasing vegetation cover the time that the soil is protected by vegetation. That not only protects the soil itself, but it decreases runoff, which is of course good for farmers, but decreasing runoff also has the environmental benefits of not polluting local watercourses. And time and again, we have seen how actions that are aimed at protecting the soil have environmental benefits as well. This last aim, I perhaps should have said first, it's really important that we develop a toolkit that is genuinely useful for farmers. If there's no balloon to take home from the party, they're not going to come to the party, right? So the way that we're trying to make sure it's useful to farmers is that we are developing it in close collaboration with farmers. For example, we conducted a survey of farmers to find out what tests they might find useful and to get their feedback on data sharing, their attitudes to data sharing. At the start of developing this, we're trialing the tests that we've developed in field with farmers and using their feedback to improve the toolkit. And in addition to co-creating it with farmers, another way to make it useful is that we're keeping an eye on ELMS and have been in touch with DEFRA. We want the toolkit to be as closely aligned as possible with ELMS reporting requirements in the future so that it can be useful to farmers in that way as well. Okay, so the 5 tests, a little bit more detail about what we're actually testing. The first one I'm going to talk about is earthworm abundance, a measure of the soil biodiversity. Participants spend 5 minutes searching for worms in a given area of soil. They record the total number, and then there's an option to separate them into juvenile and adults, and a nice key to help them then separate the adults into different functional groups, so deep living anesic worms, surface feeding worms, and soil feeding worms. And so they get a bit more of an idea of what's going on there. Doing a traditional soil texture by feel test. We've got some lovely instructions that we're putting lots of photos in so that farmers can so that participants can work their way through the test confidently. And again, in the field, we were surprised at how many farmers had never done this, and farmers were saying how it was nice for them to see how what they found in terms of the outcomes of the soil texture test tied in with what they'd heard from lab reports. We're looking at soil infiltration rate, so how quickly water is absorbed into the soil. We assess how quickly a known volume of water is absorbed into a known area of soil, and that gives an idea of how quickly water would be able to drain away during rainfall events. It's one of the more involved tests, and we've worked out that it does need some adapting, both in terms of how we describe how to do it, and also the method itself needs some change. But it was the test that farmers were most intrigued by in the field tests. Soil carbon. Carbon is obviously a very hot topic at the moment. We haven't found any reliable, simple methods for assessing carbon directly in the field. I know a lot of people are looking into this at the moment, but at present, we're using soil colour as a proxy for soil organic carbon content, using a page from the Munsell soil colour chart. And this is a method, again, that we're looking to improve. We've got some data on how colour relates to soil carbon content, but at present it's not quantitative, it's only indicative, and we think we can get it a little bit more useful for farmers in various ways. And the last method that we're looking, the last aspect of soil health that we're measuring is soil vegetation cover. We're using this lovely app called Canopio, which provides an estimate of fractional green canopy cover. So it's basically how much of the ground is covered by plants. And farmers all around the world use this. It's developed by some people in a university in Canada, and I really like it. It's simple to use. You just walk through the field for 10 seconds, I think. A little video is taken and the app extracts 10 images from that, analyzes them and pings you out a number to give you % green canopy cover. And farmers really like that one as well. So I should say that we arrived at these 5 tests from obviously extensive reading and analysis of published literature. We were looking out for methods that are cost effective, don't take too long, are simple, but can also be accurate and give results that are meaningful for farmers. Of course, we also, as I said, we oh, and I should say, yes, we did. Oh, sorry, whoops. We published a paper last year that basically summarizes available citizen science methods for for looking at soil health. So please do look that up if you're interested in it. And last point I was going to say, yes, of course, yeah, we also arrived at these 5 tests through that survey that I mentioned with farmers, and we have spoken to a lot of different soil health experts while developing this, including agronomists and scientists, as well as that farmer survey. The app will, as well as guiding people through tests, the farmers will also have some information to put these results into context. But a lot of the interpretation will come from comparing results both within what farmers have done in previous years in their own fields, and perhaps also with other farmers, using that data sharing and discussion option within the app eventually. So we don't want to provide we're not providing, this app will not provide advice. It's providing farmers with that objective data, putting it into context, but then leading them to make their own decisions about how it changes, how they might change their management practices. Okay, so what next? As I said, we are doing this in-field testing with farmers. We've done that. Some of that has been with Earthwatch staff in person with farmers to understand how they actually work with the tests, work through the test instructions. But we've also done some remotely to see how farmers get on that way. And this effort has been led by my colleague Macarena, who is also then taking what we've had as feedback from the farmers and integrating it into the tests, into the test instructions, so that we can get to the best possible place. You've heard a lot about farmers and Freshwater Watch and soil. We realise that soil and water are very closely linked, obviously, particularly in farming. We found that many farmers are interested in soil and water, and of course the two are closely linked. And we were originally going to create a separate app for the soil health toolkit, but we're now looking into integrating it within the Freshwater Watch platform, and our IT colleagues and Freshwater Watch are considering how that could work, so looking at the technical aspects of that. But once we got to a good stage, with the tests themselves, we're going to put further development on hold while we search for funding to co-develop the project further with farmers. It's really important to us that we do this properly to create, as I said, a product that is useful for farmers. One particular aspect of funding that we're looking into is working with corporate partners. We've got some interesting conversations going on with agricultural supply chain businesses, and so that's an area that we're quite hopeful that we will get some funding for in the not too distant future. So that's a whistle-stop tour of one aspect of what we're doing with farmers and soil health at Earthwatch. We've obviously talked about Freshwater Watch as well. It'll be great to hear any thoughts on what we're doing. We'd love to hear what you guys are doing and if you think there are any ways of working together. And of course, we would like to say a big thank you to funders, partners and citizen scientists and volunteers who have contributed to this work. So that's me for now. I will stop sharing my screen and then we can have any questions.